Good morning. I want to use this 1 minute to explain something that I think you didn't get it very well in the last lecture. And that is the passive tension, because I have got a lot of conversation that people cannot grasp what is the passive tension. It is very simple. The passive tension is a property of the material. Each material, if it's stretched, produce power, produce tension. Each material, it doesn't need to be muscle, and it has nothing to do with the muscle contraction. It is not the contraction force. As you see, for instance, I have a rubber band here, and it is now in a resting length. It is not a stretch. If I put it in the hand, it stays there and nothing happens with it. But if I stretch it, then it produces force. You see it, if I stretch, now this band is a stretch. Actually, and it develops force. Sorry. Here, it develops force, and with this force, actually, I can hit each one of you in the first row. Yeah, if I lose it here, you see it flies over somewhere. It is the force that it develops, and that is what I could do also with a strip of the muscle. If I stretch the muscle without any contraction, it will jump somewhere if I lose it. That is because this passive tension. The more I stretch it, the more force it develops. It is not specific for muscle, it is specific for all type of material. If you stretch, you get force. Okay, now I start with the lecture today. One other thing, I posted a few questions for you in the model. How do you call it? Module. Yeah, in the module. And then those questions from difficulty point of view comes a little bit closer to exam questions. They are not exam questions. For me, you never see my exam questions, okay? They are not in any book. They are not in any question books and so on. Also don't try to search where he gets his question. Those questions are built only by me. And therefore, these other kind of activities doesn't help you. You must understand the material. That is the point. Okay, and now to the lecture today. We have learned that if we stretch the muscle, the passive force increases, and also the active force of the muscle dependent on the stretch level. Yeah, it was the maximum at the rest condition. And I told you that this type of muscle activity, increased force or adjustment of the force, doesn't happen in the body. Because in the body, you don't stretch the muscle, you don't compress the muscle. All muscles are at the resting length, and if you contract, they produce the maximum force. Okay, so you cannot adjust the force of the muscle by stretching the muscle or by compressing the muscle. Okay, that is not a tool in a live body to adjust the force of the muscle. And there is another type thing that increases the force of the muscle, and that is the tetanic contraction. And I want to start with this. You have learned that if it is here a muscle, this muscle is innervated, and the action potential is transmitted to this muscle fiber, and then calcium is released, and then you have active mucin interaction, power stroke, and you get the muscle contraction. So here in this, you see the membrane potential. If this nerve transmits the potential on this muscle fiber, you see that is the action potential of this fiber. And this action potential continues for 1 millisecond. Also very short, 1 millisecond. And as you learn, this action potential is followed by a muscle contraction, because the action potential releases calcium and then the contraction is happening. And that is the muscle contraction. If you look at this muscle contraction continues for much longer time than the action potential. Action potential is probably 1 millisecond. Contraction, mechanical contraction is 100 milliseconds, also 100 times longer than an action potential. Okay, that is very important phenomenon to understand tetanic contraction. And now if I go to the next slide, here I give one action potential, and that is the muscle contraction which follows this first action potential, okay? Then after a while, I give another action potential, that's the second action potential, and the second action potential is followed by its own contraction, by the contraction which follows him, and that is the contraction following the second action potential. And you see that these contractions are fully separated from each other. The second contraction is started at the first one ended. Okay? These kind of contraction we call is twitches. That is a single twitch. Every one is a single contraction, a single twitch. Okay? And now I apply this second action potential, bring it a little bit closer to first. Yeah, the time difference between the two action potential, the frequency increases. And you see this one is followed by this mechanical contraction, and this second action potential produced this mechanical contraction. And now you see what is happening. Your action potential completely back to normal. But this second one, the contraction for this second one, it starts as a time that the why does this? Okay, it starts at a point that the first contraction is not ended. Okay, action potential ended, but the contraction didn't end. And then the second contraction comes and adds to the first one. You see that the amplitude of the contraction is much larger. It means the contraction force is greater. And then the third action potential comes, and again, its contraction adds to the two other ones, and you see that the entire amplitude is much, much higher. It means the force of contraction becomes bigger and bigger the more contractions are adding to each other. Please don't make a mistake. Action potential doesn't add to each other. They are all or none phenomena. Mechanical contractions are adding to each other. Okay? So, and if you increase the frequency of action potentials even further, you see all of these are the contraction which are following these action potentials. They become adding to each other, and if the frequency is very high, like here, you see, even in this case, every action potential is separated from the other. And the mechanical contraction of following all these are melting together to a smooth contraction. You even don't see the individual contractions following this action potential because they are melting together to a smooth contraction. And that is called a tetanic contraction. Okay? And you see that the force of tetanic contraction is much, much larger than a single twitch. So, now one other thing. This is also not a mechanism in the body to adjust the force of the muscle to the need. Why not? Because all the contractions that we do in the muscle by our activities are tetanic contraction. We very seldom, almost never use twitches by the muscle contraction. Every movement that I do is produced by tetanic contraction. Therefore, I cannot adjust the force of the muscle because the muscle has tetanic contraction from the beginning. If I bring you an example, for instance, if I read this, I must lift the paper and read it. If it was this condition, C, then it must be like this. Do you read a paper like this? No. So you take it in the hand and it is smooth and stable. And therefore that is the tetanic contraction by which we bring it up and we read it. You never had these twitches, single twitches. So stretching muscle, tetanic contraction, all of these are not measured to adjust the force of the muscle for the need in daily life. How do we adjust the force of the muscle then in the body? And there is one possibility for that, which is called recruiting motor neurons. What is a motor neuron? A single motor neuron is one neuron from which a number of muscle fibers are innervated. For instance, this alpha motor neuron in the spinal cord innervate, for example, these 5 muscle fibers. And these muscle fibers with this motor neuron is a motor unit. Okay? If an action potential comes down of the axon of this neuron, all these 5 will be contracting at the same time. And then their contraction will add to each other. Also, that is a force of the 5 muscle fibers. And you see here, I have 2 motor units now. One is the blue fibers are innervated with them, and the other one is red innervate the red muscle fibers. If I innervate only one motor neuron, then I have 4 muscle fibers which are contracting. And if I innervate both of those at the same time, then you see 7 muscle fibers are contracting. Therefore, the force of 7 muscle fibers is greater than the force of 4 muscle fibers. And that is the way the body works. If I want to apply more force, I recruit more motor units, okay? The more motor units I activate, the more muscle force do I get. Got it? That is the physiological way to adjust the force of the muscle for the need. And here you see, again, there's 2 motor units. I innervate the blue one, the red one. You see the red one, that's the action potential for red one, and that is the mechanical contraction for red one. And I do now, if I do the same, also this innervate the motor neuron B, then I get this action potential in the mechanical contraction of the motor unit B. The action potential remains separate, but the mechanical contraction of all these muscles add to get to each other to this one. And you see that the mechanical contraction increase is higher than the individual contraction. It is the way that the force of the muscle are adjusted in the body. So, and now you see here we have, for example, 4 units. If I innervate only 1 unit, then I get this mechanical contraction by 2 units, by 1 and 2 together, this one, 1, 2, 3, that one, and all of them together, a higher mechanical contraction. That is the way we regulate our activity the force of our muscle. And now here are examples that you see how many fibers are innervated by one motor neuron. By tibialis anterior, this muscle you know from anatomy, 450, this muscle has 450 motor neurons. It is innervated, one single muscle is innervated from 450 motor neurons. And it has about 270,000 muscle fibers. And the ratio is that one motor neuron innervates 600 muscle fibers. It means if I have one motor neuron, then I have the force of 600 muscle fibers. If I have two motor neuron, activated two motor neuron, then I have the force of 1,200 muscle fibers. And so I can go until 450 motor neurons. If I innervate all 450 motor neurons, then I get 270,000 muscle fibers and the force of all those muscle fibers together. Okay, by hand muscle, you see that this ratio of motor neuron and muscle is 1 to 100. One neuron innervates 100 muscle fibers. And by eye muscles, here, this ratio is 1 to 10. And now just tell me which of these muscles, the force of which of these muscles can be finer regulated. Finer regulated, this one or this one? Certainly the last one, the eye muscle. Why? Because I can change the force of contraction always by the force of 10 muscle, 10, 20, 30, and so on. If I do it here, I jump from 600 to 1,200. That is not a fine regulation. Therefore, always for the muscle where is needed that their force will be fine regulated, then the ratio of motor neuron to muscle fiber is much smaller, also 1 to 10. One neuron innervates 10 muscle fibers and not 600, okay? And here is a table. I don't want to go to table. It is differentiating between different type of muscle. Mostly there are three: white muscle, red muscle, and somewhere in between. And you can read this data. That is very simple. Red muscle is aerobic metabolism. It needs blood flow, and therefore is red, has more myoglobin, and it contracts energy delivery. It takes for longer, but it contracts not so fast. White muscle is anaerobically and contract very fast. And it doesn't, and the contraction doesn't take long time, also doesn't continue for a long time. Therefore, you can say the muscles which needs to produce tension for a long period of time, they are mostly red muscle fibers. They are the muscle in the back, for example, which keep your body in upright position. Okay, holding the position. They are always contracted and they have tension. That is isometric contraction producing tension that we don't fall on the ground. And other muscles which are white and anaerobically, they are fast muscle. They are used for jumping. For a short period of time, if we want to do something very fast, then the white muscle are used, okay? That is the mostly differentiation. Read the table, you can understand it easily. So, and now I go to the lecture of today, actually. One other thing, I really answer the question after the hour today because I'm behind. I must go through the lecture. Please ask me at the end of the lecture. Okay, by smooth muscle, these are the learning objectives that you read and see that they are covered. And now it is very similar, everything. Smooth muscle normally, I would say normally need for its construction also calcium. And calcium can come from outside or from inside. A skeletal muscle, it came from inside, from sarcoplasmic reticulum. Heart muscle, I told you, from outside comes a little bit, then most of the calcium is released from sarcoplasmic reticulum. But smooth muscle does everything what the skeletal muscle is able to do and what heart muscle is able to do. And even something more it can do that both of those cannot do. Okay? It is very flexible. And as I told you, one possibility to bring the calcium from outside is that the membrane depolarize. And smooth muscle fiber has also voltage-gated calcium channels. And then these voltage-gated calcium channels are opening and calcium enter the cell. That is one possibility. If there is an action potential to open this or a depolarization, it even doesn't need to be an action potential. A depolarization which can open these voltage-gated sodium channels. Other possibility is a ligand-gated calcium channel. A hormone, a transmitter binds with the receptor and opens the channel. This possibility we have seen acetylcholine, for instance, in the skeletal muscle binds with the receptors and open the channel. It is not exactly this way, but it could in some areas you find these type of channel as well. The other is a mechanical channel. For instance, you find these in the renal arterioles. You probably heard the phenomenon of autoregulation. It means if the blood pressure increases, it stretches the wall of arteriole. And as the wall is stretching, then this mechanic sensitive calcium channels are opening. And as they open calcium, the calcium comes into the smooth muscle of arteriole and causes contraction of arteriole so that the blood pressure behind the arteriole in the kidney doesn't go high or the flow doesn't increase so much. That is mechanic sensitive calcium channels. And then the smooth muscle have the possibility to release calcium from sarcoplasmic reticulum. And there you have one possibility is calcium-induced calcium release. Do you remember I told you in the heart muscle, calcium comes from outside a little bit and then opens the renin receptor to release calcium. It is the same phenomenon almost here. First, this calcium which comes from outside, open this calcium channel at the sarcoplasmic reticulum, and the calcium is released from sarcoplasmic reticulum. It is called calcium-induced calcium release. The other possibility is that the membrane has a receptor for a hormone or for a neurotransmitter. And after the hormone or neurotransmitter binds with this receptor, it activates phospholipase activity, which is in the membrane, and causes the activation of this pathway, IP3, inositol 3 phosphate. And as we have IP3 is increasing, there are calcium channels sensitive to IP3, and they are opening, and now the calcium comes out of plasma, out of his arm. You see here, hormones can produce contraction by the smooth muscle. They cannot do it by hard muscle, by skeletal muscle, and so on. By smooth muscle, because they are in different organs, and their function must be regulated. And hormones have possibility there. So there are a lot of possibilities to bring the calcium from outside or release it from inside. Okay. And now how the contraction of smooth muscle is happening. As you remember, I told you by skeletal muscle that head is ready to bind with actin. The mucin head is ready to bind, but the binding site is closed by tropomucin. And calcium must enter and do these things to open it, and then the head is binding there. So the regulation takes place at actin. Yeah, the binding site must be open to myosin. The regulation takes place there, and calcium was needed for that process. The head is always ready to bind with this. The difference by smooth muscle is, in smooth muscle, these binding sites are open. So this troponin complex is not there. And that is open, and mucin head can always bind with that binding site of actin. But it cannot bind because the head is not ready. The head of mucin is not energized enough and ready to bind with the actin binding site. Even the actin binding site is free. Okay, we must prepare the head to be able to bind to the actin. And that is what we call thick filament regulation, because mucin head is part of the thick filament. By skeletal muscle, it was thin filament regulation because it was based on actin. Here is a thick filament regulation. The mucin head must be prepared. How is the mucin head prepared for this purpose? Now, as calcium enters the cell, calcium binds with a protein which is called calmodulin. You see one molecule of calmodulin binds 4 calcium. And now this calcium calmodulin complex activates an enzyme which is called mucin light chain kinase. Do you remember my mucin light chain? I told you every head had 2 mucin light chain. One was the regularity chain to stabilize the head, the alkali chain, which stabilized the head of mucin. The other was the regulatory chain, which regulate the ATPase activity of the head. And now this regulatory chain will be phosphorylated with this mucin light chain kinase. Okay, this enzyme phosphorylate the mucin light chain, and now the head is energized and can bind with actin. It means by smooth muscle, it is not calcium directly to initiate the process of cross-bridging. It is the calcium indirectly which stimulates an enzyme, and that enzyme phosphorylates the mucin head. And now this phosphorylated mucin head can bind with the actin site. Okay? So, and after it binds with actin, the whole process of cross-bridging is exactly the same like in the skeletal muscle. Then ATP is needed to bind with the head and be partially phosphorylated, and then P go out, ADP go out, and then the next ATP binds and it's separate. Okay? In other words, for you to remember it, the cross-bridging cycle in a skeletal muscle needed calcium and ATP. And smooth muscle, it needs ATP and phosphorylation of mucin light chain. Okay? Phosphorylation of mucin light chain, not calcium directly. And now you see this slide probably you don't have, I found it 2 days ago, and therefore you don't need to have it. You see here that this calcium calmodulin activate the mucin light chain kinase, and that phosphorylate. Here is the light chain of the mucin head that is phosphorylated by this MLCK, and now the head binds ATP. This ATP is different from the ATP which is used to phosphorylate the light chain. Okay, this second ATP binds with the head and is needed for contraction, as you know. And then from there, the whole circle of contraction is happening like in the skeletal muscle. I don't need to go through that. So what does it mean? It means you have mucin light chain kinase, which use ATP to phosphorylate the mucin light chain. And after the mucin light chain is phosphorylated, then ATP is needed for contraction. The head binds ATP, and then it goes through this contraction cycle, through the cross-bridging cycle. And this contraction stops. You say by skeletal muscle, you took calcium out and the contraction stopped. Here, you must dephosphorylate the mucin light chain. The phosphorylation of mucin light chain initiated the cross-bridging, and cross-bridging stops if you dephosphorylate the mucin light chain. And then we have another enzyme which dephosphorylates the mucin light chain, and that is mucin light chain phosphatase. So this one phosphorylate and induce contraction, the other one dephosphorylate and stop contraction. So, and there is now one other difference, differentiated between phasic contraction of the smooth muscle and tonic contraction of the smooth muscle. Phasic contraction is that what I described. Mucin light chain is phosphorylated, the contraction is happening, dephosphorylated, and it stops. If the dephosphorylation, normally this dephosphorylation happens after mucin head separated from actin. After ATP is bound and it's separate from actin, then it will be dephosphorylated and then the contraction stops. But if the mucin light chain is dephosphorylated before head is separated from actin, okay, before it is separated, then the cross-bridging cycle takes much longer because head and actin remains attached from longer period. Okay, it takes longer, the force remains for longer time, but the number of cycling reduces to 30%, and therefore it uses less ATP. And that is what we call latch state. And the contraction is called tonic contraction. Okay? By tonic contraction, the contraction continues for longer time. It requires less energy, and the force is higher because the actin and mucin are connected with each other for longer period of time, and not so fast. And that is happening if the light chain is dephosphorylated before mucin separate from actin. Okay? So, when do we need this type of tonic contraction? You see, you will learn in next semester that arteriole, for example, have always a tone. They are always contracted. And by sympathetic activity, either you increase the activity or you decrease the activity. Yeah, only by sympathetic, not parasympathetic, because they are contracted already. You can increase the level of contraction and you can decrease the level of contraction. That is, this permanent contraction is a tonic contraction. Whereby phasic contraction is, and all these things which, for instance, in GI tract, as somewhere in small intestine, a contraction is happening, it is moving through all the other segments, and the first segment becomes relaxed. They are not tonic contractions, they are phasic contractions. Only that segment contracts for a certain time and relax again. Okay? That is the difference in different areas of the body. If you want to differentiate between these two forms, you see that is the calcium release in the muscle cell, in the smooth, in the skeletal. That is that is smooth muscle, but that is a phasic contraction, where normally the light chain is dephosphorylated after the contraction. And you see that here you get the calcium release, and then you get the muscle contraction, and then you have the velocity of cross-bridging cycles, and all of them return back to normal level. After a while, all of them are back to normal. If you see by tonic contraction, you see the calcium release exactly like before, but it doesn't return to normal level. It decreases to a lower level, but that lower level is higher than the normal. And then you see that the velocity of cross-bridging here get a peak, like by phasic contraction, and then it decreases. It's slowed down, the cross bridges, but it remains at a higher level and doesn't return back to normal. And the muscle force, you see, is increasing to a maximum, and it stays there at this point. Not a maximum, but at a higher level, and it stays there, doesn't return back to normal. Okay, that is a kind of muscle contraction to use less energy, in which it keeps the actin and mucin connected to each other, and then requires less energy for cross-bridging cycle. And cross-bridging cycle slowed down by this type of tonic contraction. So, and now the question is, how can we regulate the muscle force, the smooth muscle force? Want to have more force, wants to have less force. You have seen that this is an enzyme misphosphorylate, the mucin light chain, and produce contraction. This other one dephosphorylate and stop contraction or reduce contraction. Therefore, the force of the muscle depends on these two. Which of these are stronger? If I have more activity of MLCK and less activity of MLCP, increase the contraction force. If I have more stimulation of MLCK, the contraction force is increasing. If I inhibit MLCP, I reduce dephosphorylation, also the contraction force increases. If I reduce the MLCK activity, I reduce the contraction force. If I reduce the activity of MLCP, then I increase the contraction force. So it is a balance between the activity of these two. Okay, can you follow that? Okay, and now how this balance is regulated. At the part of this mucin light chain kinase MLCK, its activity, as we saw, depends on calcium calmodulin complex. If I have less calcium available, therefore less activity of this, less phosphorylation, less contraction. Okay? And there is one way to regulate the activity of MLCK. There is another way. If I phosphorylate the MLCK itself, Not that the MLCK phosphorylate the myosin light chain, but to phosphorylate the MLCK itself. And that can happen with, yeah, that is the contraction. Okay. If I phosphorylate this enzyme through cyclic IMP pathway, okay, I activate the cyclic IMP pathway, and that phosphorylate the MLCK. After MLCK is phosphorylated, has less affinity to calcium calmodulin. If it has less affinity to calcium calmodulin, then its activation would be less. And the phosphorylation here would be less, and the muscle contraction would be less. So there are two pathways to regulate the activity of MLCK. And by that, the force of the muscle. One, through calcium, increase or decrease. And the second is through phosphorylation of MLCK by cyclic IMP pathway. In phosphorylated condition, this enzyme is less active. Is it active, but less active, and therefore make less phosphorylation of light chain and produce less muscle contraction. Okay, so you have two possibilities there. Which possibilities do we have on the side of the phospholipase to regulate the activity of phospholipase? That is here. I don't want you to remember all these things. I cannot even remember if I don't see the picture, okay? But it is important for you to know which mechanisms are there to remember as you go through different organs and study that. One, you see that is myosin light chain phosphatase. How will be its activity regulated? You see here plus. Plus means that its activity will be upgraded, also stimulated. And if I follow this chain, I come to NO, nitric oxide, which are produced from endothelial cells in the vessels. You know it as a vasodilatator. Why it is vasodilatator? Because it increases the activity of phosphatase and then dephosphorylate the mucin head and then produce less contraction. If there is less contraction, then it is dilation of the vessel. Okay, therefore it is a vasodilator. And this NO, you see, after it binds the stimulate a guanylyl cyclase pathway and then activate protein kinase G, and that stimulate the mucin light chain phosphatase and reduce the dephosphorylation and increase the dephosphorylation and reduce the muscle force. Another pathway you see here through this PKC, although you can inhibit MLCP, and also through this pathway by hormones, for instance, which binds with these receptors and they stimulate this pathway, they can inhibit mucin light chain phosphatase. As they inhibit, they decrease, they increase the muscle force, because inhibition of this enzyme means less dephosphorylation. It means myosin chain become more phosphorylated and balanced to MLCK and produce more contraction force. You see, there are possibilities to increase the activity of mucin light chain phosphatase or decrease the activity of mucin light chain phosphatase. And how the smooth muscle forces depend on these two factors, okay? So, and what happens with calcium after contraction? It could be pumped out or pumped into the sarcoplasmic reticulum. You see here to the outside, you have these H+ calcium exchanger or sodium calcium exchanger and also at the wall of sarcoplasmic reticulum, you have calcium H exchanger, which bring the calcium inside this or bring the calcium out of the cell to reduce the calcium and reduce contraction. So, and now these are what you can read, how can the force will be adjusted through spontaneous electrical activity of the cell membrane, which bring calcium in the cell and increase the contraction. Neurotransmitter release could affect the muscle contraction by ligand-gated calcium channels, or by these pathways which affect the MLCP, and local factors like pH, pO, and so on. And as a stretch, we have seen if we stretch the muscle, smooth muscle cell, the mechanic sensitive calcium channels are opening, and the force will be regulated through those. So these are not new. That is a summary from what we have learned right now. And we know two types of the muscle, smooth muscle. One is called multi-unit, and the other is single unit. What is single unit? By single unit, that is right. Yeah. By single unit is that these muscle fibers are connected with each other through Gibb junction. You see, that is a Gibb junction. This is. And if I have an action potential in this smooth muscle which causes contraction, it will jump from this smooth muscle to the other and would stimulate all of them, and then all of them contract like a single part way, not individually. That is, for instance, in GI. If I say the contraction in duodenum, it goes through all the small intestine until the end, because they are connected with each other. Then single unit is the ones that individual muscle fibers are individually innervated by a nerve, and they are not connected with each other. And if I want to activate those, then I must activate each one individually. And those smooth muscles are good for fine regulation. They are mostly in iris in the eye, because it must be fine regulated if you get more light in the eye or less light. Although the iris muscle is multiple unit. Okay, what is left now for me to do to compare the skeletal muscle with smooth muscle, the mechanical properties. You have seen that is the passive tension curve, and this is the active tension curve. And that is both of them together, total. It is a skeletal muscle, that this one is smooth muscle. What is the difference? First, let's start with the passive tension. You see, by a skeletal muscle, the passive tension is resting, where you have the optimum length, where you get the maximum contraction here at the resting length, it is 0. Yeah? Normally, the skeletal muscle is not stretched. It is the passive tension is 0. As it will be a stretch, then you see the passive tension is increasing. It is different by smooth muscle. You look here at this maximum of the contraction, which is the optimal length. The optimal length of this muscle is not at 0, that is here. It means the smooth muscle is stretched. Optimum length of the smooth muscle is not a length in which it is resting. It is stretched, and then it produces more power. Why? And that is a difference, okay? And now if you look at the curve, at the active curve, you see that was one difference. The active curve, you see it is broader. By smooth muscle, it is much broader than by skeletal muscle. Why it is broader? Because it has much more, the myosin fibers, the myosin fibers in smooth muscle are longer, much longer than in a skeletal muscle. Therefore, the probability for, they give more probability to connect with active because they are longer. And therefore, the curve becomes broader. They can contract over a broader range of length. Yeah, because simply the myosin fibers are longer. And the other difference is the third one. It's kind of different, but a fact that you should remember. You see, the maximal force produced by contraction is the same by skeletal and smooth muscle. It means smooth muscle is not a weak muscle. Don't think that the smooth muscle is weaker as your arm muscle because it is in GI. It is not. At maximal force that it develops is almost the same like by a skeletal muscle. So there is in the force no difference. Okay, the force is the same. The active curve is broader because myosin fibers are longer, and here the passive force is larger at the let's say the time that the muscle is at resting, simply because it has more connective tissue and other fibers which produce the passive tension. So these three differences. If you look the force velocity relationship, this relationship is given here by skeletal muscle, and you see it by smooth muscle. Here was the maximal force because that was in isometric contraction where you produce the maximal force. And you see the maximal force for smooth muscle and the skeletal muscle is the same. You see both curves have this point together. There is no force difference. But the velocity, you see the maximal velocity is much, much lower in smooth muscle. It means the smooth muscle contract much slower than a skeletal muscle. Do you remember that this maximal velocity, it is no load? The muscle left himself depend on what? I told you yesterday. That depends on ATPase activity of the myosin head. It depends on cross-bridging of the myosin, how fast it runs. And how fast it runs depends how much ATP in a time can be break down. And that is the activity of ATPase, this enzyme which breaks ATP. And that is in smooth muscle, this activity of ATPase is much, much smaller than a skeletal muscle. And therefore, you have a much smaller maximal velocity. Sorry, that is a better picture for single unit and multi-unit, which are also phasic. Single unit are phasic contracting, and multi-unit are tonic contracting muscle. You see here that these muscles are in multi-unit, in single unit. All of them are connected to each other with Gibb junction. These are the Gibb junctions. Yeah, and the contraction starts at this place and flow over all and goes over all these muscles, like in GI tract. But you see here that that these muscle fibers are not connected with each other through Gibb junctions, and each one is innervated individually. So they must be activated individually, and that is the case by Eris muscle, and that is called multi-units, and the contraction is mostly tonic contraction in those muscles. So that's all. Thank you very much. That is all what I have with you.